Is this a pointer? No. This is a pointer. Wait, I will need to point. Ahí está. Ignacio Sanchez and myself established a lab like 10 years ago, and now we are fortunate enough to work with Ezequiel, which is right here. He will answer all your questions in the poster session, and we collaborate with Ernesto, which is close by in a laboratory. So you probably know already about proteins. Everybody should have seen a structure of a protein. It's really amazing, such an amazing structural forms, right? These are all structurally resolved high-resolution X-ray crystallography of different proteins. And the amazing thing, for some of us, is that they are just made of 20 amino acids. It's really impressive that you can make these fantastic objects just with 20 amino acids. In 1957, the founder of molecular biology, Francis Crick, proposed what is called known as the central dogma of molecular biology. He says that once the information has got into a protein, it can never get out again. So he established these arrows that show the transfer of information from one molecule to another one, but never from protein to protein or back to this. I claim and I state here that we should stop teaching this. This is terrible. This is not true, and this is dangerous, because in popular understanding of science, this can lead us, okay, we shouldn't care about what happens here, everything is here. So all the information for making an organism is in the blueprint of the genome. You probably heard about this. Why South Americans play so good soccer? Oh, it's in their DNA. Oh, why do they have so much trouble in Argentina? Oh, we have the Peronist genes. I see this in the actual popular press, in the newspapers, in the television, right? So we now have the DNA as the sole culprit of everything. Well, but this is simply not true. Why? Genomes are not stable entities. We know this. The biosphere, we can call that it's a big evolutionary experiment. Apparently, the N is 1 because all of the biosphere apparently has a single origin 3.8 billion years ago. So if you look at the amino acids of related proteins, you see that they are related somehow. And the fact is that the functional patterns of the proteins modulate their genomic sequences. The biological functions are emergent from proteins in a medium. So we should restate the flow of information from sequences given an environment and typically in microsecond to second to structures. This is called the folding problem. These structures are not unique. They are molecules. They are 300 Kelvin. They should move. And it's apparent that the dynamics of the proteins, which usually takes place in the millisecond to the gigasecond timescales, give rise to chemical activities. And these chemical activities, together again with a given environment and in these timescales, give rise to biological functions. And the fact is that we have a feedback loop from the biological functions to the sequences by processes of evolution. Usually these take place in the terasecond to the petasecond timescales, but there are some examples in which you have evolution in the gigasecond timescales. We should point out that the information here is digital in the sequences, but analog in the rest of the information flow. We should also put a break here that this is equilibrium thermodynamics, and this is out of equilibrium thermodynamics. And as Chris Adami pointed out in 2015, we should think of evolution as a process where information is flowing from the environments into the genomes. Right? This is Lamarckian, classically Lamarckian. But I agree with it. It's not Lamarckian is not a bad word. Nowadays, we find out that a lot of mechanisms seems to be Lamarckian. So, But this is a very imprecise statement of information, right? We just put arrows. What is really information in these places? So we can quantify information as the Shannon uncertainty before and after, right? This is just we can quantify the bits of information before and after. In the case of sequences, we can quantify the information before is in a random polypeptide minus the evolved polypeptide, which is related to the alphabet number in the random sequences, or the alphabet number in the evolved sequences. And in the structures, we can quantify the information as the change in uncertainty between the unfolded state and the native state, which is related to the number of effective conformations in the unfolded state and the effective conformations in the native state. We have done this for a lot of different ways of measuring, of approximating these numbers, and for the sequences or for the structures, which we call Levinthal information gain. And it happens that the average of these numbers is very similar. There are about 2 bits per site per operation in evolved sequences, and there are about 2.4 plus minus 0.3, again, bytes per site per operation in evolved structures. So, on average, the sequences contain just enough information to code for folding, which implies that there is little room to code for something else, which is the function of the protein, which is the one that is actually selected. If you code something else for function in the protein, it might conflict the folding. And this has been seen in a lot of computational studies. When you evaluate how good sequences fits a structure, you can find out that the places where it doesn't fit very well are related to the functional sites, allosteric sites, catalytic sites, binding sites, and so forth. So folding must be a very strong evolutionary constraint. Where does the alphabet size come from? We have a very simple relationship. If the information in the sequences is close to the information in the structure, this is just related to the effective conformations in the unfolded state on the native state, and the alphabet size and the evolve per site alphabet size. So this gives us this beautiful relationship. And at the limit, when the number of characters in the digital characters in the evolved sequences matches the number of effective conformations in the unfolded states, and we approximate any native to 1, we have that the alphabet size is related to the number of conformations in the unfolded state squared, which should be equal to the number of effective letters in the evolved sequences squared. So we have this relationship. This is a theoretical relationship between the alphabet size and these two characteristics. We just have 2 data points. Proteins have an alphabet size of 20, and the evolved effective size is here, plus minus the different estimations. And then we have another foldable and evolvable polymer, which is RNA. RNA structures can also be coded in linear strings of nucleotides, and here is the evolved the evolved measurement of the effective evolved characters in these polynucleotides. And then we have also the unfolded states. Number of conformations in the unfolded states for proteins is around here. It matches the theoretical observation, theoretical prediction, and for RNA, it also is in the range. So, biopolymer chemistry, which is the one that determines the number of effective conformations in the unfolded states, constrains both the alphabet size and the effective digital size. So we ask, Okay, there is information there to code for folding. How does the change in the sequences modulate protein folding? We want to predict the folding mechanism, the overall stability, cooperativity, intermediates, and so, only using sequence information. And we want to do it on a large scale. So we propose a general scheme. We take a large MSA. MSA is multiple sequence alignment. We basically take sequences of organisms we take the sequences of homologous proteins of different organisms, we align them, we infer some evolutionary energy. You probably heard about this DCA in the class of Morcos, right? We infer an evolutionary energy, and we apply it to a toy folding model in which we coarse-grain the amino acid sites to icing spin. Some can claim that we are analyzing the proteins of the spherical cow that you physicists love so much. Yes, we will analyze spherical cow proteins. Basically, again, we take the MSA observables, the frequency at each side, the pair frequencies, and we construct a pot slice model in which the energy of the sequences is determined by a local field, which is the propensity of each amino acid at each position, and the coupling between the amino acids, pair amino acids at pair positions. We do this with a Boltzmann learning. You can do it with a restricted Boltzmann machine, and now you can do it with a large language model and whatever, so you know much more how to do this. The thing is that this fits and it's convergent. So how do we map the evolutionary energy to a folding model? Okay, suppose we have a sequence here of a natural protein. So we divide the protein in what we call foldons, or elements, right? You physicists love the electron as the one that has the electronic charge, the neutron as the one that has neutron, the codon as the one that codes. Well, here we have the foldon, is the one that folds. The foldon is defined for a given part of the sequence, and the foldon can be either folded or unfolded. Here is the Hamiltonian. If the foldon is folded, you have an internal energy of the foldon. If the foldon is unfolded, this contributes with an entropy. If two consecutive foldons are folded, then you have a surface energy. So this is a typical icing spin, right? You probably know much more about this. So for every foldon, we have an interaction energies that are determined by the sequence. But how do we determine this? Well, we know from the DCA the couplings, the energetic couplings for every pair of amino acids. We just get, okay, for this foldon, we just sum up all the interactions in this foldon. For the interactions between foldons, we sum up the couplings between foldons, and so forth. So basically, it's a mapping of a POTS model to an icing model over proposed foldons. So in this case, we did it for repeat proteins. In repeat proteins, this is much easier because the foldons are easier to define. When you're talking about globular proteins, well, the foldons, you don't know where to put the boundaries between the foldons. What we will use is the exon positions. Probably you know that eukaryotic organisms do not have the genes contiguous in sequence, but they are spread in parts that are then spliced out. The introns are spliced out, and then you end up with a mature messenger. So if you look at the position of the exon boundaries, you see there is not a flat distribution, but there are exon boundary peaks, which we call minimal common exons. So this is the boundaries we will use to define the foldons. We learn the evolutionary energy for the whole family, and we project it to these internal energy and interaction energies, and we do an icing simulation. For this, it is indispensable to have the selection temperature, which is the scaling between the pots and the icing. So basically, this apparent selection temperature, it can be calculated from experiment, because you can make a mutation and see how much the free energy changes in experiment, and then you can measure the change in the energy for the computational model, and the slope of this will be related to the selection temperatures. Even assuming that all families have the same delta-delta G distribution, this can just be scaled from the variance in the energy of the Potts fields. And then you do a simulation, a Monte Carlo simulation. This is the fraction folded as you raise the temperature. Everything is folded at low temperature. As you increase the temperature, this thing unfolds. But it doesn't unfold all at the same time. You see, there are different parts of the protein unfold at different times. This is the structure, the zero temperature structure of this particular protein. And we can compute the folding free energy change as you change temperature. Everything is unfolded. As you lower the temperature, a free energy barrier happens, and you get a minimum at 5 folded elements, which you can then propagate down. You can compute the correlation matrix between the folding of the elements. So you see that this barrier really corresponds as folding 5 elements, which are 1, 2, 3, 5, and 6. So this green, yellow area is this particular yellow area. And then you fold this, and then you fold this. These apparent subdomains are compatible, qualitatively compatible with experimental XC exchange and molecular dynamics simulations. Then, since this is a very simple mapping, we have thousands of sequences. So we can run the simulations for different families, from different sequences, from different families. And we can analyze, well, how does the sequence changes in each family impact on the folding variations. This is the overall view of the whole data set. We simulated over 7,000 sequences. This is the distribution of the folding temperature for the different families. Some families have a low variation in the folding temperature and a higher mean folding temperature. Some families are very spread in their folding temperature. We found that the variation in the folding temperature is basically related to the selection temperature, to the T cell. So the higher the T cell, the more variations that are allowed in the sequences. And those variations impact on the folding mechanisms. We should point out that all the families are within the energy landscape theory limits. In energy landscape theory relates the fallenness of the energy landscape as the ratio between the folding temperature and the glass transition temperature. I don't know if you've been taught this in Morcos classes. Anyway, don't worry. It's how funneled the energy landscape is. And this is the relationship between the selection temperature and the glass transition temperature, which is basically related to the evolutionary optimization of those sequences. Everything is between the limits set by energy landscape theory, fortunately. And we can analyze how is the cooperativity variations. Not all domains fold with the same cooperativity. Some of them are all or none. Here, for example, these two things are the heterogeneity in the energy of the interacting elements. Basically, if the fold-ons are all equal, the heterogeneity is 0. And this is the interaction energy between the fold-ons. So if the interaction energy between the fold-ons is 0, then we have there. The whole family of tryptophan synthase A doesn't spread very much in this parameter space, and everything is very cooperativity. Color here is cooperativity, 1 is complete cooperativity, 0 is non-cooperativity at all. So some families, no matter how you change the sequences, you really don't change the folding cooperativity because you're not changing the heterogeneity of the interactions or the surface between the interactions. Then you have some other families in which the different sequences spread in this plane. And you see that the color smoothly changes from very cooperative to non-cooperative. The very cooperative is when you have low variation in heterogeneity and high interaction energy. Basically, everything collapses altogether. And if the things are more different in the internal energy and they don't have enough interaction energy, then you have a non-cooperative folding. So, but you see that some families are sensitive to the cooperativity transition, and some families are sensitive, and some families are not sensitive to the transition. And this is not related to the selection temperature. What is related to is basically to the topology. We know there are proteins that are just composed of alpha helices. Some proteins are composed of just beta strands. Some other proteins are composed of both alpha helices and beta strands. And you can compute basically the number of contacts in the structure that are short range or that are long range. And this is related, basically, the alpha helices are, most of them are long, dominated by short-range interactions, and they have a high variance in the cooperativity. And basically, the beta folds are more dominated by long interactions, and they have a lower cooperativity variance. You can model this with a pure topological model, and more or less the distribution is sustained. And with this, you can calculate the impact of the point mutants. So here is the position in the sequence, and here is what would happen to the cooperativity when you change one amino acid position for this other, and how you change the folding temperature when you change every amino acid in every position. This is basically like a deep mutational scanning of a particular sequence. And again, we can do this in seconds. So we can explain that the cooperativity and the folding temperature variations are related to the evolutionary energy, but unfortunately, we have no high-throughput folding experiments to compare these two. They should soon come out. So, on average, sequences contain just enough information to code for folding. The amount of information in the sequences is very close to the amount of information needed to specify a fold. There is little room to code for function without conflicting folding. Folding is a very strong evolutionary constraint. And I propose here that biopolymer chemistry is the one that constrains alphabet size. And this is why we have 4 nucleotides and 20 amino acids. Sequence variations impact on the folding mechanisms. The folding temperature variations are related to the selection temperature, and the cooperativity variations are constrained by the overall topology of the system. I think we're done.
So you have time for a few questions?
Thank you for your very nice talk. My question is, today we know that there are some proteins that are non-structured regions, and maybe your idea to define a structure, this what we call folding. Maybe it will be nice to investigate this kind, because they say that it's unfolded, but it's not really totally unfolded.
Exactly. Well, it's difficult.
Can you go in there?
So it's difficult to work with intrinsically disordered proteins. So at the first, I will call that all the measurements we are doing and all the statistical analysis we are doing it over globular proteins. Right. Those are the 70% of the genomes of eukaryotes and are about more than 90% of the genomes of prokaryotes. So intrinsically disordered proteins are very difficult to work with because you cannot get alignments. It's very difficult to align the system. It's very difficult to experimentally validate how folding they are because they are very sensitive to the environment. You change a little bit the salt, you change a little bit the pH, and they completely explore different structural forms. And they also undergo multimerization, liquid-liquid phase separation. So
But yes.
And they are complete. So some of them, you can treat them thermodynamically reversible cases, but most of them, it's apparent that they are not reversible transitions. So.
But even in globular protein, I don't know, you look just to the interaction between amino acids, but there are some physical-chemical entropy there.
Yes.
Does not consider.
Yes, I haven't talked about this, you are right. So these are completely sequence-based potentials. We have no chemistry, we don't know nothing about the interaction of amino acids, we don't care about this. We have to care about this is where the entropy terms comes in, right? Where do we get this entropy? This is a free parameter. So we fitted this free parameter with the experimental folding curves. So for some proteins for which there is a reversible thermal transition, we have the transition, we make the simulations at different parameter, we explore the parameter range of this entropy, and we fix it. And it turns out it's about 5 kcals per mole per Kelvin, which is reasonably in what people have measured. But we take this as a single thing, and we applied it. We fit it for one family, and we applied it to every family. It's a single residue entropy. Yeah, of course, but this depends on temperature also. So it's I mean, we're talking about the spherical cows. I know about this, but we can learn something about the spherical cows also.
So very nice talk. I really enjoyed it. But I think kind of following up on Paulo's question, in addition to the environment that drives folding, of course, as we know, the interaction with other proteins and with lipids and whatever other molecules. But I was thinking that most of the proteins that are in the 15 families that you studied probably live their lives in the cell as multimers. And they're not necessarily monomeric, right? They work as dimers, trimers, tetramers.
Not particularly. We took care of not to put dimers, tetramers, and so.
I noticed, for example, that in the one slide that you showed with the in which you highlighted tryptophan synthase, for example, there was triose phosphatidylisomerase as well next to it. Those are all multimers, like dimers.
Triose,
yeah. Tim,
I think it's Tim barrels.
Tim
barrels.
Just the barrel.
Yeah, it's just a barrel, single barrel.
Single barrel?
Yeah.
But that doesn't exist in nature, does it?
Well, so we have a problem here.
Yeah.
In general, I see we have a problem, which is saying what is a protein family and what is not a protein family. It's the same problem biologists have in defining what is a species and what is not a species. This is subspecies. So we have a definition of what a protein family is. We took a practical definition is what PFAM says is a family. That's it. Or what Interpro now says it's a family. Whatever they construct as a family, we just take this, we take the hidden Markov model, and with that we scan the databases and construct the multiple sequence alignment. Of course, this can be populated by many subfamilies. Some of them might be dimeric, some of them might be monomeric. Yeah, it's noisy. It's noisy. We haven't looked at if some of them that change are particularly monomeric or dimeric. We haven't done that analysis. You're right.
We have time for a last question over there.
Okay, I had two questions, but I think for the sake of time, I'll only ask one. It's really incredible. I was thinking if you have looked to proteins of thermophiles, if there is any particular signature for this temperature folding?
Yes. In general, this is not something new. When you learn these evolutionary fields, it just happens that if you do an approximation of the folding temperature for different sequences from different organisms from which you know that they live at different temperatures, it just happens that there is a correlation between the evolutionary energy that you calculate and the actual living temperature of the organism. This is well known for a couple of examples for some systems, and it's impressive, no, in itself. Because remember, we are building this assuming that the sequences are in equilibrium, which they are not, of course they are not. But the assumption that they are in equilibrium and we can learn an evolutionary field assuming that they are in equilibrium, well, it seems to relate to some biological phenomena. That's really impressive for me.
Fantastic. So let's thank our speaker again.
Thank you.